won't have many time for questions, so that after the talks people can join there and interact with the speakers. So thanks Phil once again, and now I think we can move on to the flash talks.
I think, can you maybe announce the next flash talk?
Yes, I thought, yes, I can. I was just waiting for the video to be set up. So the video is ready to be set up. So next talk, the next talk will be by Siddharth Jayaraman from the Roslin Institute. He will be talking to us about the Bovine Omics Atlas. Please, Siddharth, please. We can start Siddharth's video.
Hello. I would like to use this opportunity to present my work on Bovine Omics Atlas, BOmA. BOmA is an online graphical genome browser to visualize omics dataset across bovid species. I'm Siddharth, and I work for James Prendergast's group at Roslin Institute, and I'm developing and maintaining BOma's backend and working towards adding new feature sets. Our aim with BOmA is to develop an online resource to inform future improvement of species like cattle and water buffalo and allow cross-species comparison of omics data. BOmA provides resource on selective sweep, structural variants, and chromatin data across multiple cattle and water buffalo breeds. Our cattle browser presents data on 5 cattle reference genomes and an expanded reference genome augmented with sections of the pan genome not present in the original reference. BOmA presents the first high-quality assemblies of African cattle breeds of Ankoli and Endama, and the manuscript describing the dataset presented here was recently submitted by our group. Cattle browser features tracks containing attack-seq peaks to assess the genome-wide chromatin accessibility, cross-breed comparison between assemblies, structural variant calls made using BioNano optical mapping datasets, and variants called using short-read sequencing. To access these datasets, please follow the link to the browser for your breed of interest, and on the top left, you will find a link to open a track selector. Once in the track selector view, select the dataset you wish to view and return to the browser view to dynamically interact with the dataset. Apart from the cattle browser, BOmA includes a water buffalo genome browser. In South Asian regions, water buffalo is an important source of milk more so than cattle. The data presented in BOmA has been recently published, and our browser provides genetic variants called from whole genome sequencing data of 7 water buffalo breeds. And selective sweeps combination presented by XPEHH and XPCLR scores are available for water buffalo breed pairs and 13 cattle breeds lifted over to their orthologous locations in water buffalo genome. Just like the cattle browser, please follow the link to the water buffalo browser and you will be presented with a very similar interface, and you can pick and choose the dataset to view using the track selector. BOmA is currently under active development, and I'm looking into adding new datasets which are currently being analyzed across different livestock species. The eagle-eyed viewers among you would have noticed that BOmA is using JBrowse to visualize the dataset. We chose JBrowse because of its ease of use to set up and is light and configurable. Just 1 week before this presentation, JBrowse announced their latest release, JBrowse 2, with some significant changes and improvements to visualization, along with some new features. We believe BOmA will benefit from these new visualization feature sets, and I'm currently trialing a version of BOmA with JBrowse 2 and will be ready for release in coming weeks. This work wouldn't have been possible without the help from members of James Prendergast's group, especially James, Andrea, and Preston, who did all the data analysis work, and Elena for making the BOmA logo. And of course, our funders at Roslin, Edinburgh University, CTLGH, and BBSRC. I would like to thank you all for your attention and happy to take questions and feedback for future developments. Thank you.
thank you. Thank you for your talk. It was very pragmatic and objective, and pretty much on time. So we have time for questions.
There is ongoing work to have multiple reference for the bovine because the species are different, breeds are different, have different genomes. How it's going to be taken into account in your system?
So at the moment, we have different browser for each of the different breeds. But in the manuscript, we are actually developing. Andrea from our group generated a pan genome, like a graph genome for the cattle genomes. So that's all coming soon. So at the moment, you can pick and choose whichever breed you are especially interested in.
Thank you.
One question. Do you take in only for de novo genome data or also resequencing data?
So we have 2 new genomes. So that's kind of what was assembled from our group. But for the other genomes, it's been taken from the RefSeq submitted sequences.
So it's resequencing data.
Yes.
So if the community, someone in the community generates WGS to characterize and study other breeds of the world, it would be interesting, and you could upload in your atlas.
Yeah, we are working. So even within Roslin, we are working with other groups. So probably in near future, we may have a African buffalo added to the BOmA browser as well. So we are looking forward to adding more species going forward.
Okay, thank you.
Thanks.
So I think if we don't have more questions, I would propose that we move on to the next speaker. Next speaker is Bala Kiran Manthri from SLU University, and he's going to talk to us about structural variations in the cattle genomes.
Hello all. Thank you for everyone for giving me this opportunity, and I'd like to thank my supervisor Thomas and my friend Fanitra for being with me during this. My name is Bala Kiran, and I'll be doing my master's in data science, part of computer science. Today my topic is finding structural variations in the dairy cattle. Our aim of this project is to find the best computational approach for identifying structural variations and finding the best evaluating software for a number of individuals should be sequenced for a long read sequencing. Our aim is to, and we would like to build a graph genome with all the identified structural variations. Uh, we can say that impact of structural variations on a genome expression is known to be very significant, but the estimation of the structural variations would be very difficult. And in this project, we would like to use long read sequencing, which is a basic step for finding the structural variations. And we have been used Holstein and Jersey cows as a part of research. And all the information which is being taken from a giga cow, which is an internal project of SLU. Uh, let's talk about what is the structural variations. Uh, it is defined as a region of DNA more than 1 kb, which has been which has inversions, deletions, or translocations, uh, and duplications, all any other. And we can say it as we can call it as a copy number variation as well. Uh, Okay, our aim is to build a graph genome. Like graph genome have been constructed from a population of genome sequencing such that each haploid genome in this population is represented by a sequence path through the graph. Let's see this example. We have a reference genome on the top, and uh, we have uh, we can consider the below genome genome sequence, which if we see we can consider a single nucleotide polymorphism in the left side, and if in the if the T missing over there, we can be considered as a deletion, and if the A is added to the sequencing, we can say it is an insertion. Uh, Once the graph genome is built, it has been indexed through specialized tools. This is the sequencing reads are then aligned to the graph, which is more accurate than alignment of reads to the single reference genome. The alignment are then cooperated and stored. In this way, the graph genome is used to call all the structural variations. These structural variations can give us a better understanding in the cattle. Like we can understand how healthy is the cattle, how how it can cure itself when it is infected with any virus or something, how it behave with others, how it behave with other individuals in the cattle, how it is different from other cattle. So all these things we would like to find out all these things in the cattle for better uh, cattle milk production or beef production. Thank you.
Andrea, you're muted.
Sorry. Hi, sorry. It's so many buttons, the video and the and and the microphone. So I was just saying that we'll be able to take some questions for Bala regarding his work. So I have one question.
Yes.
Bala, are you there?
Yeah.
Okay. So you are going to be considering just this small panel of breeds, or do you intend to enlarge your
Actually,
breed sampling?
in the first part, we try to consider very little breeds, and then we want to increase the samples actually.
Mm-hmm. Okay.
Yeah.
Thank you. So we may move on to the following talk, which is entitled Detection of Mobile Genetic Elements Insertion Sites in Swiss Cattle Breeds. This talk will be given by Meenu Bhati from the TH Institute in Zurich.
Hello everyone, my name is Meenu Bhati. I'm a doctoral student at ETH Zurich, working under the supervision of Hubert Pausch. Currently, I'm visiting the Giga Institute of Leibniz University, and currently I'm working on detection of mobile genetic element insertion sites in the Swiss cattle breeds under the supervision of Carol Charlier, and this work is a part of BOVReg work package 2 and 4. So what are these mobile genetic elements? These are also known as transposable elements, which are the DNA sequences which have ability to change their position within a genome. And they contribute almost 47% in the cattle genome. But not all these MGs are still active. And this can be divided into the two major classes, retrotransposons, which are LTR and non-LTR, and DNA transposons, which are not active. So why we are interested in these MGE? Because some of the MGE retrotransposons are still active in cattle, and they contribute significant source of de novo mutations. Also, their insertion can cause genomic rearrangements. Also, they can affect the expression pattern, either modulating the regulatory sequences or the expression, or they can also create genes and RNAs. So our goal is to establish the catalog of the polymorphic MG insertion sites in the Swiss cattle breeds. And in this one, I'm using 3 Swiss cattle breeds: brown Swiss, original brown wheat, and gravy data. So how we can identify these insertion sites? Here is a toy example. In a candidate DNA or an individual in which the MG is inserted, you can see there are the 2 green boxes. They are actually the identical sequences which are present on the both side. And when we sequence the genome of this animal with the paired read ends, and when we align these reads against the reference genome, which doesn't have MG insertion, these reads will be aligned like this. Here you can see some read at this point would be showing the split reads, which is similar to MG but not present in the reference genome. And there would be other reads, which is known as discordant read, in which the one mate is aligned to other part of the reference genome, which are similar to these sequences. So the target site duplication, split read, and discordant reads are the signatures of MGE. So in my current work, I'm using a locator pipeline, which was developed by the former PhD student at GIGA, Chad Harlan, and currently with the help of Leaching, the current PhD student, I'm establishing this pipeline in the Swiss cattle breeds. This pipeline basically identified the insertion sites for LTR and line elements. And the important step can be divided into the 3 major steps, in which the input is BAM files. In the first step, we try to identify the insertion sites per sample. And this one, the criteria is it need to have at least 3 discordant reads, both on forward and reverse side, at least 1 split reads, and the mapping quality of those reads should be about 20. So basically, we are using high coverage samples. In the second step, we are merging these data for the all possible carrier animals. To refine all these insertion sites, once we identify on a high coverage samples considering the requirements, we can genotype them also in the low coverage samples. And for genotype, you can see if the TE insertion is absent, it is known as homozygous 0. When TE insertion is present just in one haplotype, it is known as heterozygous TE insertion. And when the TE insertion is present in the both haplotypes, it is known as the homozygous TE insertion. And currently, I'm in the second step of working on the second step, merging of these insertion sites in my data set. So how we can visualize these one with the BAM files. So this is the IGV view of the 3 cattles. Here you can see, this is the top line is the coverage, and these are the aligned reads. And exactly at this position, there are 6 base pair target site duplication. You can see double the coverage, which are, so on this breakpoint, there are split reads on both sides. And they are also covered by multiple colorful reads, which are actually the discordant read pairs. So it is present in all the reads. So it is considered as a homozygous TE insertion. Whereas in the heterozygous, where you can see in the second animal, it is heterozygous based on there are few reads. So the gray reads, which has no discordant reads, which are properly aligning to the reference. So these are properly aligning at this T insertion site, which is showing that one haplotype does not carry this insertion. Whereas in the last third animal, this is a homozygous reference or missing. You cannot see any split reads or discordant reads in this sample. So further, once we identified, and the leaching also realized that the many polymorphic insertion sites are very recent and they are low in frequency. So it's very difficult to impute them to genotype. So we're going to use these identified insertion site on a custom genotyping array to genotype them on a population level. Further, the effect of these insertion sites will be associated with the several phenotypes and eQTL in a work package 4. That is from my side. Thank you.
You have to unmute yourself, Andrea.
Sorry. Sorry. My timer, it seems. Cancel. Sorry, my timer was not, didn't want it to be canceled. So thank you very much, Siddharth. It's quite a challenge that that you have working with mobile elements. Is there any questions to Siddharth? To Meenu, I'm sorry.
I was confused for a moment.
Oh, sorry. No. So I have, yeah. So it's I have, so the way that you identify the reads that that can be from MGE. So you have the discordant reads and you have the split reads. Could you explain this a bit better?
Okay, so at a certain position, if a MGE sites are inserted, so those elements are absent in the reference genome, but exactly where they inserted, usually they cause the target site duplication, which I showed them by green boxes. I could not explain that more in 6 minutes, but those green boxes actually are the identical sequences which are signature of MGE. So when a MGE inserted in a genome, they causes that site to be duplicated on both sides. So when that MGE element is not present in the reference, so you will see exactly on those green points, which are only present as a once in genome, you will see all those sites causing a split reads and discordant reads.
And do you have plans to make a de novo assembly for these samples where you detect the MG insertions to validate the? So then you would have like the complete sequence of the animal, because now you're aligning to a reference genome which doesn't have the MGEs.
Yes, but the reference genome also consists those MG positions somewhere else.
Yes.
So which are similar to the bovine one. So once you identify, so using the custom genotyping array, you can validate them if they are actually present or not. So that's the further part.
Okay, thank you very much, Meenu.
You're welcome.
So we'll have now our last talk.
Andrea, there is one question more from Kylie. She just raised her hand.
Oh, sorry. I think we have time, right? Please make your question.
Two quick questions.
Kylie, can you speak a bit louder, because it's...
Yes, can you hear me now?
Yeah, now it's much better. Thank you.
Excellent. Thanks for your talk, Meenu. With identifying these MGEs, presumably the reference sequence has some in them, but not all of them, not all the new ones.
Yeah.
Is your method able to identify only the new ones or the existing ones and genotype at the existing ones as well?
By far, what I have understand in my last 1.5 month stay, which I'm currently at Liège. So currently we are only can focus if those sequences are already present in the reference genome. So if those MG insertions are already present, so those transposable elements are already described in the using the repeat masker and RAP-based database. So if something is totally new, it would be difficult. But if MG element contained from a similar family, So you would be still able to extract them, or can say that this MG might belong to this one, but not totally the new one. So I hope I able to explain.
Yeah, mostly. Thank you. And then just finally, with your mapping, are you using soft clipping options?
Yes, actually,
Yes.
I did not say that. Those split reads are actually the soft clip reads, and
Yeah.
the program also take the hard clip sites into the consideration.
Great. Thank you very much.
Thank you.
So I don't see any raising hands now. Are there? No. So I think we can move on to our final flash talk of today, which will be entitled GWAS, mQTL and eQTL studies with imputed whole genome variants for BovReg traits. This talk will be by Praveen Krishna, and he's from Leibniz Institute for Farm Animal at FBN.
Thank you very much.
Dear Nextflow workshop members, thank you for accepting my eQTL presentation for the flash talk. This is Praveen Krishna Chitneedi, and I'm working in Institute of Genome Biology under Dr. Krista Kuhn. And we work with Holstein and Chevrolet cattle and pure Holstein cattle from FBN. And I'm also a member of BOVREG project. And in this project, I'm working on integrative genotype-phenotype data analysis of work package 4. In this project, I already performed analysis of genotype imputation and GWAS. For the imputation, I performed stepwise imputation using Beagle version 5 and GWAS using GCTA. In future analysis of this project, I'm going to work on eQTL studies, and also there is a possibility to implement this pipeline in Nextflow. Coming to my previous work, I updated the SNP coordinates and also the reference and alternate alleles of the genotypes in accordance with the whole genome sequencing. And then I performed genotype imputation using an iterative masking strategy. And after this strategy, we got around 99% to 96% in different steps of imputation. And for the GWAS, we used mixed linear model-based association analysis using GCTA software. And the input file for this software is ML dosage and ML info format. And these are the prepared using imputed genotypes from 29 autosomal chromosomes. And the phenotypes include beef trait, milk production trait, feed efficiency, and metabolites. As the genotypes are imputed, we use dosage values in order to address the imputation inaccuracies. This is one of the example of our GWAS results. From beef trait carcass and meat quality, we used around 19,59036 from biallelic traits. We got some significant SNPs SNPs across genome in different traits related to this phenotype. And in relation to eQTL analysis, this is basically a GWAS using multiple phenotypes like gene expression, exon expression, and other expression phenotypes. And this can be broadly divided into two types: cis eQTL, which influences the nearby genes, and trans eQTLs, which influences the genes which are far away from it. For example, genes in different chromosomes. Coming to BOVREG project, our goal is to perform a single or multi-trait association test using imputed whole genome sequence variants and read cones from different groups. And for this, we need to develop and standardize a pipeline. And there are some requirements to implement eQTL analysis. The first is sample size. It is required to have a large number of sample size to identify gene expression and associated loci variability. And also the quantification at right age and physiological state with good number of read counts is required. And eQTL allele specific expression requires unambiguous phase genotypes for all the heterozygous variants. And also it is required to have a unique definition for molecular phenotypes when the data is collected from different sources, so that in order to have a harmonization across read quality control, alignment, and read counts. And there are some challenges to implement eQTL analysis. The first is, in case of multi-breed eQTL study, some of the specific eQTLs related to a breed might be overshadowed when the breed size is comparatively smaller, and when there is a multi-tissue eQTL detection, the heterogeneity of the multi-tissue have different effects, which presents a key challenge to detect eQTLs. And it is required to distinguish allele-specific expression of cis eQTL from the allele-specific expression of parent of origin or imprinting. And it is required to distinguish cis eQTL from trans eQTLs. For example, some of the local eQTLs which are generally considered as cis eQTLs, but sometimes they can be trans. My main motivation to join this workshop is to develop a Nextflow work pipeline to harmonize RNA-seq phenotype count matrices across different partner groups from BovReg, as this reduces the bias to process the samples from different groups for the eQTL study. As the eQTL pipeline was already developed using Nextflow workframe for the human species, which is called as eQTL catalog, I am planning to develop a similar pipeline for the cattle genome using Nextflow. Thank you for your attention.
Thank you, Praveen. Praveen, you are here for taking questions?
Yeah, of course I'm here.
Okay, good. So trying to see if there are any raised hands.
I have a question.
There is one from Cedric.
Go ahead with the other one. Go ahead with the other one.
Daniel Fisher, your hand is raised.
But I can wait until I was the last, I suppose. Go ahead.
Okay, in that case, this was just a question out of curiosity. I'm thrilled to hear that you're going to use Nextflow. How do you feel about NF-Core? So this morning you had the first overview of NF-Core. How do you feel about exploring the connectivity between your pipeline and NF-Core? And possibly is there proposing a new type of pipeline to NF-Core, or incorporating the benefits of NF-Core? Is it something you've already looked into or not yet?
Yeah, I already looked into this thing, and yeah, actually currently I'm exploring the different permutations and combinations for using different tools, which can tailor made for my pipeline, and based on these tools, I will compare the pipeline which is already existing, and then I will decide how to proceed further. So it's not at all decided yet, but yeah, in the coming days I will. Yeah.
And what you, because comparing tools is not complete, is not simple, and that's actually true for all the pipeline we heard about.
Yeah.
Are you exploring different type of metrics? Like one, of course, is efficiency, but there is a problem of accuracy and a problem of actually generating your own metrics for accuracy. So is it something you've looked at yet, or?
No, actually, I have yet to receive the data. So once I receive the data, I will check the matrices and I will come to a conclusion how to proceed further.
Okay, thank you. I see a comment of Phil to everyone that there have been discussions about new GWAS pipeline, imputation, and eQTS analysis in NF-Core, and then and that but there is a GWAS channel, GWAS Slack channel on NF-Core. Okay, so and there is you've seen. Do you have? Is there anything published on that stuff yet? Is asking Zainab to everyone, to you. Is there anything published yet on your pipeline?
No. It's not yet published. It's still all the work is still going on, and once it's finished, actually the GWAS and imputation is finished, but we are still waiting for the eQTL analysis. Once everything is ready, maybe we will be going to publish. Yeah.
Thank you. There are other questions that I'll leave it to Merit. Oops.
Daniel?
Hello. That is maybe a rather specific question, but for the eQTL analysis, the association tests, do you know what kind of tests was done there? Is it a linear model based, or what kind of eQTL test do you do there?
Actually, in the different studies which I studied, there are following different approaches, mostly based on the linear mixing model, and sometimes also they are using regressions analysis, and it's not like a specific way like GWAS or RNA-seq, but there are different approaches. So I have to decide what to choose based on the data. Yeah.
Okay, so it's not decided yet. Then what is the?
Yeah.
Praveen, I have one question. So you, in the end of your talk, you mentioned that there is a pipeline for humans and for analysis of eQTLs.
Yeah.
What was the main points of divergence that you found between this pipeline and now that you want to apply to bovine?
I think the main difference would be, I can say, like the sample size, and also how to handle the read count matrices. In case of bovine and human, it's quite different. So I have to check how they can be comparable, and whether I can adopt the same pipeline as it is, or maybe I can do some variations and permutations to this using different tools. For example, I checked another paper where they use leaf cutter for slicing of RNAs, but in this catalog they used something else. So I have to check how it can be compared, and I can use it or not.
Yeah. Okay. Thank you.
Hello there. Can you hear me?
Yes.
Yes, I just have a quick question. You mentioned in the slides about some challenge with the eQTL analysis, and it was interesting to ask whether you have any plans with the breed-specific eQTL, because that seems to be a very interesting thing.
Yeah, actually, in the BOvReg project, we have data from different breeds, and also we have our own breeds. Like one is like pure Holstein and Holstein Chevrolet crossbreed. So we will be exploring first with that across different breeds, and then in the further studies we'll be concentrating on our own breeds. So there is a possibility. Yeah.
Oh, good. Thank you very much for that.
Yeah.
So this was Tuan, that it was you asking the question, right?
Yes.
Okay. I don't see any raising hands anymore. I'm trying to browse through all the participants. So I would like to thank everybody for participating in this session. And I would like to remember that there is one breakout room for each of the flash talks, and one break room, which is called Introduction of NF Core. So where anyone who wants to join can interact and make questions to Phil. So I will ask all the speakers to join into the breakout rooms, and all the participants to just, if you have more questions or anything else that you would like to discuss, to join there.
Sorry, there is one more announcement by Jose before you guys leave.
Yeah.
Yes. So yes, just to mention that we will start the hackathon at 2:30 in a joint session before we going into the breakout rooms for each project. So this was not in the schedule, but it's now updated. And the idea is to see in this session which are the advanced and objectives of all the projects. And this way someone can rethink to join a different project. And if we have someone joining us, because I had also some requests, this person could know what are the projects about. So those presenting try to be around some minutes before the starting of the session. And as I told you, all of you, I was in all the breakout room sessions of the projects. It shouldn't be something very exhaustive. It's just so that people see what are you working on and which are the people that are participating. So I think, yes, that's all from my side. And see you. I encourage you to join the breakout rooms now and see you at 2:30.
And can I ask the organizers if we can meet on room 7 right now? Just have a 5 minutes meeting on room 7 if you're free. And thank you all. This was once more a great session. Thank you, everybody.
Thank you. And see you tomorrow on streaming on YouTube again.